And we have this exam coming up, and you've learned a lot of stuff so far. And you might look at everything you've learned so far and be like, okay, this is a pretty good immune system. Why do we need the rest of the semester? Why is it that the things we've learned aren't enough? Is everything that we've learned thus far sufficient for immune protection? Or are there any big gaping holes at this point? So the one thing that we've learned about thus far is phagocytosis, where we've seen a microbe get phagocytosed by something like a macrophage, as is shown here, or a neutrophil, and get degraded inside of the phagosome. That seems like a pretty effective mechanism. However, there are some microbes that have evolved ways to survive inside of a cell after phagocytosis. This includes microbes like Listeria monocytogenes that actually breaks a hole through the phagosome and escapes and lives happily ever after in the cytoplasm, getting lots of nutrients from us and in a nice protected, stable environment, and they're real happy. There are other microbes that can either prevent fusion of that phagosome with a lysosome, so we never get an acidic environment, we never get all of the degrading enzymes, any of that stuff. There are others who can actually modify that phagolysosome, again, to make sure that the pH doesn't get terribly acidic, to not have the degradatory enzymes, any of that kind of stuff. These are the strategies that are used by things like Salmonella and things like Mycobacterium tuberculosis. And so in these cases, phagocytosis is definitely not going to deal with the microbe. The microbe wins if phagocytosis is all we have in these cases. We can also think about antibodies and whether or not antibodies are sufficient for immune protection. Here you can see some antibodies binding to a virus and blocking that virus from entering a cell. What do we call this antibody function again? Neutralization. So we can see a nice example of antibodies neutralizing a virus. And this seems like a pretty good mechanism to deal with viruses. Well, here is a cartoon example of a virus, HIV. If we have an antibody that's going to attack HIV, it is going to be able to get to this yellow protein, GP120, pretty easily. It might get to this red protein, GP41, pretty easily. But the antibody can't go through the viral membrane and attach to internal proteins. And so, in fact, this mechanism of antibody surveillance is sort of blind to the inside of the virus. It's really only going to detect the outside. And if you think about the airport, just looking at the outside of things is not frequently considered sufficient for protection, because something bad could be on the inside. And so the antibody has this flaw that it's not going to be able to look at any of the internal components of a microbe. The reason why this is important is that a microbe like HIV could have some mutations to change its outer layer. As is shown here, it can go from a purple outer layer protein to a red outer layer protein to a blue outer layer protein, but it could leave everything inside intact and thus avoid antibody recognition. So in this case, we could have changes to the outside of the microbe, avoiding antibody recognition, just leave the inside the same. And again, antibodies are really out of luck here because the antibody can't actually recognize the internal components of that microbe. And so that's a pretty good reason why antibodies are clearly not going to be sufficient. The other reason why antibodies are not sufficient also has to do with viruses. You might realize that I like viruses, so that's why I'm giving you virus examples. So here you can see the HIV replication cycle. You can see HIV getting into cells and then performing all of its steps of replication, and this cell is one of your cells. Antibodies would be awesome at combating that HIV virus when it is outside of the cell. It would be able to neutralize that virus beautifully. But once the virus gets into one of these parts of its replication cycle inside of cells, the antibody is useless. The antibody can't go into your cells and start to combat the virus. So antibodies are really good for extracellular pathogens, but once the pathogen is inside of cells, they're kind of useless. They can't go into the middle of your cells and start to destroy things. And if you sort of think about virus replication as is shown here, the virus is inside of cells the vast majority of its life cycle. For most of the time, it's doing something inside of cells. And so antibodies are useless in those points. They're only useful to keep the virus out at the beginning. And you could think about some of our antimicrobial peptides in complement in the same way. They're going to be pretty useful when a microbe is outside of cells. They're not going to do much for microbes that are inside of cells. As a result of these problems, hopefully what you see here is that antibodies and phagocytosis and the things that we've learned about are not sufficient for immune protection. There's still some big missing pieces. And one thing that a lot of these missing pieces have in common that I've shown you is that they involve something being problematic on the insides. So we have an inability to detect what's going on inside of the infected cell. On the previous slide, you saw the inability to detect targets that are on the inside of a virus. With the phagocytosis example, you saw inability to do something about those bacteria once they are inside and have been phagocytosed in the cell and have modified the cell. And so this other arm of the immune response that we're going to talk about is largely responsible for dealing with things that are inside of cells, or looking at the components that in fact are on the inside of pathogens. And so that's kind of the big distinction that we're going to see here. These responses are all mediated by T cells. T cells are a kind of lymphocyte, so they are part of the adaptive immune system. They're going to undergo VEJ recombination. They're going to do a lot of the same types of things that we saw with B cells. One thing to be aware of with T cells is that T cells are subdivided quite a bit into many subpopulations. Today I'm going to largely talk about T cells as a whole. I'm not going to subdivide them too much, but in the future we're going to be subdividing our T cells up quite a bit. So T cells are really going to be the key responders that you see here. It's hard to give you a definition of a T cell. With a B cell, it's easy. I can say a B cell is a cell that makes antibodies. It's an antibody-producing cell. It's much harder to do that for T cells because there are so many types of T cells. And it's hard to make a definition that fits all the types. This is a figure depicting some T cell responses from your book. And it shows you two major types of T cells. So here you can see the B cell, and you can see that it's producing antibodies. Here we've got our two major types of T cells. One of them is going to be able to kill an infected cell. The other is going to produce a lot of cytokines that can act on other immune cells to help their responses. They're going to be the helper cells and the killer cells. The only definition I've ever seen that really describes the action of all T cells together is that T cells are cells that interact with other cells of your body and induce them to change. So the killer T cell interacts with another cell of your body and induces it to change by killing it. The helper T cell interacts with another cell of your body by producing cytokines and helps it to improve its responses. But really getting a good definition of T cells will vary based on the T cell subset that we're talking about and the way that those individual cells work. So your B cell had an important receptor on its surface. What is the name of that receptor? It is the B cell receptor. So it turns out the T cell is going to have an important receptor on its surface too. What do you think is going to be the name of that? The T cell receptor, exactly. So our T cells have this important receptor, the T cell receptor. We've talked a lot about the structure of an antibody thus far. We've seen that the antibody has a heavy chain and a light chain that pair together. They have a variable region and a constant region. And at the very tips of their variable region, there's an antigen binding site. You might remember that I told you about the FAB portion of an antibody, which is sort of the individual arm that can be made with different digestions. It turns out that the T cell receptor basically looks like an FAB. So it's got two chains. One of them is a heavy chain, one of them is a light chain. They're held together by a disulfide bond. We've got a variable region and a constant region. And the very ends are the antigen binding sites. They're going to have 3 CDRs. And they're going to have loops that touch the antigen and all of that kind of stuff that we've seen before. So it's going to be, it's actually really, really similar to an FAB portion of an antibody, but there is a transmembrane domain that allows this to be a receptor. So the receptor looks pretty familiar. If you look back at the course schedule and the things that we have covered thus far, the way I covered B cells, you will notice that first I introduced you to the general idea of an antibody. And then I took a little detour and told you about antigens and some of the important properties of antigens. And once we knew about antigens, we came back and talked about antibodies and B cell development and all of that business, right? We are going to follow that same scheme for T cells. So I've just introduced you to the T cell receptor. And we are now going to take a little side jaunt into thinking about the antigen that the T cell receptor binds, just like before we talked about the antigen the B cell receptor binds. The difference here is that the antigen is way more complicated. And so, in fact, we are going to be talking about this antigen today and all of next week. And so it might feel like we're doing this in a weird way, but it's actually the same structure. It's just that we need to spend some serious time on this antigen. The antigen was first described in a specific experiment. You can find this experiment in figure 7-4 of your textbook. I'm using this version from another textbook because I like the way they've used terms a little bit better. And so I think this one's slightly more straightforward, but you can find the exact same experiment in figure 7-4 of your textbook. This experiment was done by 2 investigators, Rolf Zinkernagel and Peter Doherty. They won the Nobel Prize for what they've done in this experiment. They are still alive. They are active scientists. I follow Peter Doherty on Twitter. So they were the ones who really started to understand the antigen for T cells based on this experiment. In this experiment, they had a few strains of mice. Remember that a strain of mouse is a group of inbred mice, all of whom are genetically identical. In some places, they have strain A mice. In other places, they have strain B mice. They first took a mouse from strain A and infected it with a virus. Officially, it's a virus called LCMV, but you don't really care about that right now. And they assumed that this would induce the mice to make some T cells and activate those T cells in response to LCMV. And in fact, 7 days after infecting the mice, they isolated T cells. Really, they isolated splenocytes, just like you did in lab a few weeks ago. So they took those T cells, and they were the killer type of T cells. They then set up another group of cells that were known as the target cells. And ideally, a killer cell will kill its target. Some of the target cells were also from a strain A mouse. Some of the target cells were from a mouse of a different genetic background, strain B. In both cases that are shown here, the target cells are infected with LCMV as well. The same virus it was infected we had from this other mouse. In some of the conditions I'm going to show you later, we can also see target cells that are uninfected or we can see target cells that are infected with a different virus, like influenza. So those are sort of the variables here. They mixed these cells together in a dish, as is shown here, and they performed an assay known as a cytotoxicity assay. I'm going to show you the classic way of doing a cytotoxicity assay. They are not particularly fun. People have been trying to come up with all sorts of new fancy modified ways, so if you end up doing immunology research, you may never have to do a cytotoxicity assay like this. And if that is the case, lucky you, because they're not really very fun. But this is sort of the classic way it works. This one usually is one that's pretty easy to wrap your head around. So we start with our target cells. The target cells were the cells that were on the right-hand side here. They're the ones that we hope are going to get killed. And we label those cells with radioactive chromium. So we keep those cells in the presence of radioactive chromium. There will be lots of radioactive chromium inside of those cells after the labeling. And in fact, it's not really shown here, but then you wash off the excess. So there's only radioactive chromium inside the cells. There isn't any more outside the cells. You washed off the excess. You then add your killer T cell that you hope should kill the target. If the killer T cell does kill the target, suddenly your radioactive chromium is released into the media. And you can very simply just measure how much radioactivity is in the media. And so the more active the killer T cells are, the more killing happens, the more radioactivity you're going to see in this assay. So Zinkernagel and Doherty set up this experiment. They always used T cells that came from a strain A mouse that had been infected with LCMV. And they varied what was going on with the target cells. They found that if they had, so again, strain A T cell from a mouse that's strain A and infected with LCMV, if they use target cells that are from strain A, so the same genetic background, and that were infected with the same virus, then they got killing. The experiment worked. The CTLs, the T cells, the killer T cells were able to kill. If they use those same T cells from a strain A mouse that had been infected with LCMV and mix them with strain B cells that had been infected with LCMV, the killing didn't happen. In fact, there was something about this T cell response that was related to the host's genetics. There was something going on here where the T cells knew what was self-MHC, or what was sort of self-cells for this killing, what was the self-strain. If we use target cells that were not infected, then those T cells also would not kill. And that one kind of makes sense, because you could assume the antigen isn't there. There's no infection. It's not shown here, but if we used a strain A target cell that had been infected with influenza, that also would not work. We would also not see killing. And so when we see a T cell response, there seems to be this part that's coming from the pathogen, where you have to have the right pathogen. That's important. But there's also this host genetics piece. The target cell has to be from the right strain of mouse. If you use the wrong strain of mouse, even with the right pathogen, the experiment doesn't work. And so this showed them that there was something about the host genetics that was involved with the antigen here, and that was involved with making this work. What we now know is that T cells and the T cell receptor see a very different antigen than B cells and the B cell receptor. B cells see free antigen. Or B cell receptor sees free antigen. Just binds to the B cell receptor, and we're done. With T cells, the antigen is actually a combination of two things. One of them is a piece that comes from the microbe. It is a peptide. What you might get from this is only proteins do this T cell response. T cells generally only respond to proteins, not to other things. So we have to have a peptide in addition to this self protein, this host protein that is listed here as MHC. And so the T cell's antigen is a combination of the peptide from the microbe as well as the host MHC. Thus, our T cells are what are known as MHC restricted. So the T cell is able to bind to both the MHC molecule, which is this molecule that's shown in yellow, and the pathogen peptide. If the MHC does not match, here you see a green MHC, peptide matches, it's the same peptide, but MHC is different, it doesn't work. And if you have the right MHC but the wrong peptide, the T cell recognition doesn't work as well. And so our T cells are restricted in terms of what types of MHC they are able to see. There are a couple ways to think about what MHC is doing. Dr. Miller and I talk about MHC in a slightly different way. The way she talks about it with her non-majors and the way I talk about it with you guys. So here you can see a broad overview of the function of MHC. We can see that there is a protein from some type of pathogen in our cell. That protein will get degraded by proteolysis, and some fragment of that protein will get placed onto the MHC molecule to be presented to T cells. And so the T cell will bind to this combination of the MHC plus peptide. Dr. Miller likes to think of this as being a little 4-year-old with a trophy who says, look what I got, look what I got, look what I got, and shows off the things that it caught. We're going to talk a little bit more about things like proteins that aren't from pathogens and stuff like that, so we need to be a little more subtle than just things we caught. And so I think about it as like a flagpole that's on the surface of the cell, where it's basically showing these are the things that are inside of me to the outside world. And so the MHC is, you can almost think of as like a scaffold that's showing off the peptides. It can either be a trophy of the thing you caught, or just some peptides that show you're healthy and happy and are okay. But they're all being presented on this MHC scaffold. One thing that's very different here than with the B cells and the B cell antigen that you saw is that first and foremost, MHC only presents peptide. With the B cell antigen, you could have a nucleic acid, you could have a carbohydrate, you could have a protein, or you could have a lipid. MHC only presents peptides. And so you largely are only going to see a T cell response to a protein antigen. With the B cell, we also talked about how the antigen remained folded in most cases. Remember how we talked about, like, if you did your Western blot and you denatured, you could denature your antigen and then everything would be terrible and your Western blot would fail and you'd be very sad. With T cells, in fact, MHC is presenting a linear string of amino acids. It's a linear part of the peptide. So the peptide can be, or the protein can be folded, but before MHC actually gets part of it, the peptide is going to be broken down, and a linear string of amino acids, a string of amino acids that are together in the primary sequence of that protein, will be presented on the MHC, as is shown here, to be recognized by the T cell. This will be a short peptide. Some of its amino acids will be important for binding the peptide into the MHC's binding pocket. So the MHC has this nice binding pocket. Some of the amino acids, shown here as shapes, will be really important for making sure that the peptide stays bound to the MHC. Other parts of the peptide will be recognized by the T cell receptor, and the T cell receptor will also recognize portions of the MHC protein. So it's really one big surface as far as the T cell is concerned. Yes, Nadia? Yep. Mm-hmm. So that's a great question. So your T cell is restricted, MHC restricted, in that it can only respond to your MHC. It's restricted to only respond to yours. It can't respond to Cal's or Malik's, only yours. Yep, Molly.
Going off of that,
Mm-hmm.
how many different types of MHC are there if every single person receives
4 slides. Good question. It's coming up. This is just sort of another view of that same picture of MHC T-cell interaction. So this is first a diagram where you can see the 2 chains. This is basically a heavy chain and a light chain of the T-cell receptor. You can see that there are some 3 little loops here that are coming down to touch MHC plus peptide. Here's the MHC molecule, here's the peptide. So you can see that interaction. This is the same thing shown as a cartoon. Here you are the T cell looking down on the MHC plus peptide. So this blobby thing in black is your view as the T cell. The yellow here is the peptide. All this around is the MHC. And so you can see that the T cell is binding across both the peptide and the MHC. That's also shown here. The gray box is the T cell's footprint. The yellow and green are the MHC. And so you can see that the T cell is binding quite broadly across both the peptide and the MHC molecule. In fact, the angle of binding that is depicted here is actually quite important. Sometimes one thing that happens with autoimmune interactions is that the T cell binds at the wrong angle onto an MHC molecule. Yeah, Malik?
It binds to the beta sheet portion?
It binds actually to the alpha helix portion. The beta, it's not shown here super well, and we're gonna get into that a little bit more. But the beta sheets are almost like a platform and there are alpha helices across the top of them, and we'll see that more. So just to give you a general overview, on Monday, today I'm going to tell you a teensy bit about MHC genetics. On Monday, I'm going to tell you a lot about MHC genetics. I'm also going to tell you a lot about the structure of the MHC molecule. And so you're going to understand things like where those beta sheets are, where the alpha helix is, and what exactly these structures are. So that's going to be Monday. Wednesday and Friday, we are going to go into detail of this stuff. How exactly the pathogen proteins are degraded, how they're put on the MHC molecule, so how we get all of these parts of the process to happen. So that's Wednesday and Friday. But today we're going to talk about just a teensy bit about MHC genetics so that we can get into some interesting stuff about MHC. So the human MHC, which is also known as HLA, but that's a nomenclature thing for Monday, is found on the short arm of human chromosome 6. MHC is very important for transplants and transplant rejection. I am always able to remember that this is on chromosome 6 because Michael Crichton wrote a book called Chromosome 6, where the idea was that he was going to make primates that had organs that could be transplanted into humans. So he gave the primates the human short arm of chromosome 6, and apparently it also made them sentient, and it's real crazy. But it's called chromosome 6, and so I can always remember this is on chromosome 6. And if you look at the HLA region, first of all, it's huge. So it's 4,000 kb, so 4 million base pairs. It's really, really, really big. And you can see that it is divided up into 3 regions. A class 1 region, a class 2 region, and a class 3 region. The class 1 region encodes genes that are called the MHC class 1 genes. The class 2 region encodes genes that are known as the MHC class 2. And the class 3 region encodes a bunch of genes that are important in the immune system, but they're not MHC. It just so happens that they're in the same area. One thing that's in the class 3 region is all of the complement proteins. Also the cytokine TNF, all of which are encoded in the MHC class 3 region. I'm going to go into this more on Monday, but I will just point out that one of the first big distinctions we're going to have to see is this distinction between class 1 and class 2 MHC. For MHC class 1, you have 3 different class 1 genes. And I'm giving you human nomenclature now. We'll deal with the wonderfulness of mouse nomenclature on Monday. Your human class 1 genes are A, B, and C. Your class 2 genes are DP, DQ, and DR. One thing that you might notice is that class 1 genes have 1 letter, and class 2 genes have 2 letters, thus hopefully making it easier to understand and read what's going on. Because, as it's shown here, the human MHC is also known as HLA. Typically, those are known as HLA-A, HLA-B, HLA-C, but again, that's a Monday problem. There's one thing that's super different about these MHC genes compared to, say, the antibody genes that you saw, or some other types of genes. Your MHC genes are co-dominantly expressed. Here you can see chromosome 6 from mom and chromosome 6 from dad. Mom's chromosome 6 has an A, a B, and a C. Dad's chromosome 6 has an A, a B, and a C. Yeah, I can see. Yeah, they're not in alphabetical order because that would just be too easy. You can see that mom has a DP, DQ, and DR. Dad has a DP, DQ, and DR. On the surface of these cells, all of these proteins are made and produced. And so you can see that mom makes A1, B1, and C1. Dad makes A2, B2, and C2. And so the surface of the cell has all 6 of those class 1s made on the surface. No MHC type is dominant over the other. You make them all. You make all those proteins. Similarly, you make all the class 2s as well. And so you can see that here. So this sort of goes along with Molly's question. Here I'm showing that mom and dad have totally different class 1s and class 2s. I kind of mentioned with my answer to Nadia's question that her T cells are restricted to her MHC, which is different than Cal or Malik's. And I said that there's something going on with MHC and transplants. So you might start to notice there's something going on with MHC and like diversity between people. Before I show you the next slide, I want to ask you a question. Let's estimate for a second that if I sequenced the DNA from all of you that encodes your actin gene, how many different versions of the actin gene do you think I would find in this class? What do you think, Cal? Cal thinks 1. Why do you think 1, Cal? It's conserved, it's important. If your actin got messed up, you get messed up. So probably everybody has the same actin gene. There's probably like 1 allele of actin. I mean, yes, there probably are like a couple that are other ones that are associated with disease, but most everybody has 1 allele of actin. Most genes, we have a very small number of alleles in the human population. MHC genes are the most polymorphic genes in the human population. So we have the largest number of alleles. So for example, the B locus, HLA-B type of class 1, there are 3,221 different versions of HLA-B within the human population. This is current as of January 2017. You can actually go to this website that's listed in your book if you'd like to see today's number, because we just keep on finding new ones. You can see how many HLA-As, how many HLA-Cs. So you can see that there are huge numbers of these. In fact, it seems as though we have not had natural selection get rid of mutant alleles. Instead, natural selection has kept mutant alleles in MHC to give us this great diversity. So here, it seems like having really diverse MHC must be a good thing for us. Having diverse actin is a bad thing for us because then you can't make cells. But having diverse MHC seems to be good. This is known as a type of selection that's known as balancing selection, where we have selection for diversity. This is important for a bunch of reasons. One reason why this is really important is because typically when people do a lot of types of genetic testing, they can test for MHC types and get a really good idea of who you are. If we do your particular combination of HLA-A, HLA-B, HLA-C from your mom and your dad is likely quite unique to you. And so a lot of types of HLA testing, or a lot of types of genetic testing involve looking at your HLA genes. Yep?
Would it be the same for like identical twins?
For identical twins, it would, yes, because they're coming from the same original egg and sperm. But for fraternal twins, it would not. Yep, Vinny.
So if you get one from your mom, one from your dad, what about when you pass it on?
Great question. So I'm going to come to that in just a second. And that's actually kind of related to the next point I'm making. So one of the other words that comes up when we talk about MHC is a genetics term. And this genetics term is known as the haplotype, is a haplotype. And I will admit I had not encountered the term haplotype until I started thinking about immunology and MHC, but it is really a broad genetics term. A haplotype is a group of genes that are inherited together. There is rarely recombination between them. So you tend to always get that block of genes together. So the MHC region of the human genome is the longest haplotype in the human genome. It's this set of genes that are very frequently inherited together. To give you an idea of, try to make that more clear, the second longest haplotype in the human genome is the haplotype that encodes the genes for hair color, eye color, and skin color. So you know that frequently pale skin, blue eyes, and blonde hair go together, but it's not 100% of the time. Every so often they get different, because sometimes you get pale skin, blue eyes, and not blonde hair. But you also can sort of recognize that there are patterns that pretty frequently go together with these things. And so that's the second longest haplotype. These go together even more frequently. As a result, you can actually think about inheritance of MHC pretty easily. And so we can do things like paternity testing with MHC pretty easily because we know some things about MHC haplotypes. So what you can see is some data here where we have offspring, biological mother, and 3 potential fathers. And this is basically where somebody did some sequencing, we'll say. There are a few different ways you could do this. And they found that the offspring had type 3 HLA-A and type 43. And B54 and B59 and HLA-C5 and HLA-C8. Sometimes you will see questions like this on an old exam. And you will see that what I ask you for is I ask you some questions about naming the HLA haplotypes. What I'm basically asking you for is which HLA genes are inherited together. And so, Vinny, you would basically inherit either one set of chromosomes. You wouldn't have recombination between the two. And so it will look something like what you see here. So let me show you how I would figure out the HLA haplotypes here. I'm going to figure out the HLA haplotypes for the offspring and for the mother. And then I'm going to ask you to tell me who's the dad. So if I look at the offspring and the mother, I know that the offspring must have received HLA-A3 from the mom, because A3 is the one that they have in common with the mom. So they have one chromosome that has A3 and one chromosome that has A11. This one came from their mom. Oh, sorry. This one came from their mom. This should be 43. 43 came from the dad. Then we can look and we can say, okay, so they clearly got B59 from their mom, because that's the one they share with their mom. And so B59 has to be on the same chromosome as A3, because they traveled together from the mom. They were inherited together. And B54 had to be on this chromosome. And then we can see, okay, well, the child definitely got C8 from their mom, because mom only has C8. So C8 must be on the same chromosome with A3 and B59, and C5 must be on the other chromosome. So these would be the two haplotypes of the child. Mom's haplotype, one of hers is A3, B59, C8. Mom's other one is A11, B78, and C8. That would be mom's other haplotype. So I can name mom's haplotype, I can name the kid's haplotype. Like I can name both of moms, I can name both of the kids, I can name whatever. But we could also use this to do paternity testing. So who's the dad? Father 2, I hear like a resounding consensus. How do we know it's father 2? Yes, Molly. Okay, so he has A43 as an option. He has a B54. He has C5. He's the only one who could have passed on all three of those. And so we can use the knowledge that MHC genes are inherited as haplotypes in this way. Yep, neat.
So does this mean siblings who are not twins would also have the same?
They could. It's a probability thing. Did they both get, the same one from mom? Did they both get the same one from dad? So it's a probability question. Okay, so the other thing that you might think a little bit about is about why in the world is it a good thing that we have this level of MHC diversity? And in order to help explain that, I've got to tell you a little bit about that diversity. If you were to take all of the different MHC molecules that are out there and try to figure out where are they the same and where are they different, where is this diversity in this MHC molecule? It turns out they're not diverse all the way across the board. They're actually pretty similar for most of the molecule, and there's one place where they're diverse. And the place where they're diverse is shown in red. The amino acids are dotted, or it's just shown in red on these cartoons. And what you will notice is the place where different MHC molecules are diverse is in their peptide binding region. So each MHC type is able to bind to a slightly different peptide. You can think of that on this figure as well. This green MHC type has a pocket with a shape that binds little roundish amino acid and binds to triangle amino acid. And it binds to a roundish amino acid at like number 2. And this one at like, I don't know, number 7 or something, whatever. And you can imagine if I drew a different MHC type, it might have a different shaped pocket. Otherwise, it looks exactly the same. The only place it's going to vary is in this pocket. And it might bind to amino acids in different locations of the peptide, or it might bind to different amino acids. So maybe it binds to like, I don't know, star amino acid in the middle or something. So you can imagine that's how each of these many different types of MHCs vary from one another. What that means is that it means a couple things. First of all, if you look at what you see here, only some parts of the peptide are contacted by the MHC molecule. And so each MHC molecule doesn't really just bind to only one type of peptide. It binds to one similar group of peptides. So for example, this MHC molecule binds to peptides that have a phenylalanine or a tyrosine in position 5 and a leucine or an isoleucine at position 8. And so all sorts of other variants, peptides can bind there as long as they have those two motifs. So those would be kind of the little pocket shapes for that kind of MHC. And so you can see that this is able to give a nice response to this virus Sendai virus, to this virus VSV, to LCMV, to influenza, to hepatitis B. All of them can induce a response with this MHC type. And with this MHC type, it's position 2, you need a tyrosine, and position 9, you need a valine or isoleucine. So it's a different kind of pocket, and you're going to have a good response to influenza. So happily, influenza is working for both of them. And this one's going to have a nice response to Plasmodium yelli and Plasmodium bergii, which are 2 parasites that can cause things like malaria. So you can see different microbes can be combated here. And different MHC types can lead to better or worse responses to any particular pathogen. So this table shows a bunch of MHC types and how they impact HIV progression. Does a patient have a slow progression to AIDS, or do they have a fast progression to AIDS? Do they have a high amount of virus or a low amount of virus? And so you can see that MHC type sort of varies in terms of how well you're going to respond to this virus. This becomes really important with the idea that your MHC genes are co-dominantly expressed. So with your MHC class 1, for example, you have 6 MHC class 1 molecules on the surface of all of your cells right now. And so you have 6 kinds of pocket that can present antigens, present peptides from your pathogens to start a T cell response. So you have a set of 6, and somebody else has a set of 6, and somebody else has a set of 6. Ideally, between those 6, you should be able to do something against most pathogens. Although maybe you make a really strong response, like in the HIV example, where a person with B27 has really slow progression, because B27 helps you make a super good response. And so maybe you have something that's good for that particular pathogen. Maybe you have something that's not so good for it, like maybe you have B58, which leads to a rapid progression. But you got 6 chances. So you should have something for each microbe. This is why we have this selection for diversity. This is great for us as a population. It might suck for you as an individual. You might have the wrong MHC type for some pathogen that you get infected with, and you might get really sick. But somebody in the human population has the right MHC type for it, has the MHC type that makes an awesome response. And so because we have this super diverse set of MHCs, somebody is going to live through whatever pathogen infection happens. That's why it's great for us selection-wise to have diversity. That's why having lots of different MHCs has kept happening during evolution, because there's no one MHC type that allows everybody to die or everybody to live. So diversity has been maintained through the population. So yeah, it could be bad for you. You could have the bad type for whatever pathogen you have. But you could also have the good type. And so for a population, this is awesome for us to have this many alleles. Yep, Cal. So Cal wants to know if you see geographic differences, and the answer is in fact yes. So there is pretty big diversity globally in terms of predominant HLA types. This actually makes things like transplantation even more complicated. If you think about how you're going to find someone with a match for you for transplantation, trying to find someone who has matching types can be important. Most of this is probably due to genetic drift, although you could theoretically think about past pathogen exposures in different areas, but it's probably genetic drift. There is a famous anthropologist named Cavalli Sforza. He actually died earlier, I believe this week or last week, really recently. He used MHC data to prove a hypothesis where there were a bunch of people on a Pacific island, and there was this question of, did they come from Asia or did they come from South America? And by looking at MHC types, he was able to actually get an answer there. And it was like a really important thing anthropologically that he was able to do. So the other thing that you might think about with this is it's really advantageous for you to have super diverse MHCs, right? Like, you want to be like this kid who has 2 different A's, an A3 and an A43, one from mom and one from dad, and 2 different B's from mom and dad, and 2 different C's. You don't want to have 2 with the same A and 2 with the same B and 2 with the same C. Then you're kind of losing out in diversity. Then you only have 3 pocket types instead of 6, right? That could be bad. How could you make sure that your child, your offspring, has the best possible MHC diversity? Yeah, Molly. So you should have a kid with somebody with different MHC types as you. That way your child is going to have super diverse MHC types. Do you tend to, on Tinder, ask people their MHC type? No, right? And so, like, well, that makes a lot of logical sense. You can't actually figure that out, right? Marina, do you have a question? Oh, yeah. Okay. So it makes some logical sense. That would be nice, right? But sort of biochemically, you're like, okay, how does this work? That doesn't make any sense. I usually, when I ran this class in the spring, this was always right around Valentine's Day. And so this became really funny to talk about on Valentine's Day. Somebody did this experiment. It's known as the sweaty t-shirt experiment. I am not making this up. They had a bunch of men. They had a bunch of people who were first checked for their MHC type. And they had a bunch of male students who wore a t-shirt for 2 consecutive nights. There's another version of this experiment where they had to work out in the t-shirt. And then they gave it to the women, and they asked them which one smelled the best and who they thought was the best smelling. And it turned out they seemed to be really attracted to the ones with the most diverse MHC type from theirs. And so you might look at this and be like, okay, this is nuts. And this is sort of, doesn't necessarily have great molecular biology mechanism behind it. If I had to guess what was going on, because you cannot smell somebody's MHC type, my guess is that your MHC type is altering your microbiome, and you can smell somebody's microbiome, potentially, is my guess about what's going on, though obviously no one has proven anything like that. So, next time on Valentine's Day, you just can't figure out what it is about that person. Maybe it's because they have really different MHC than you. Yes, Molly.
How hard is it to find out what your MHC is?
So the original way that people did it was using antibodies against the different MHCs. Now, basically, you sequence. And just do sequencing and find it out that way. I had actually considered that as a semester-long lab project, but there are reasons to not go into that, or that we don't do that. But I had actually thought about that. And one of the reasons why I worry about telling you about things like that is because there are different disease associations with different HLA types. And so you wouldn't want to find out your MHC type and then realize, oh, I have this really high chance of getting this terrible autoimmune disease. So most autoimmune diseases, for example, are particularly linked to HLA types. So there are some, like ankylosing spondylitis, where the patients always have this one HLA type, B27. There are also things like type 1 diabetes, where the idea is just that you never have DQ6. So the variation goes in both ways. And this is also kind of a pros and cons thing. B27 is actually the best MHC type for HIV progression. But you might get ankylosing spondylitis. So every MHC type has some good and some bad to it. Fortunately, we as a population all have slightly different ones. That's going to give us all a slightly different response. So one of the reasons why I would not want to do this in class is, well, then what happens when we self-diagnose with some potential big HLA problem. This is a short list of diseases that have been associated with MHC type. And what I will also say about this is very frequently people do these big, many million-dollar genetic studies where they try to find the gene associated with some disease. And oftentimes they will get this paper and their answer will be MHC. And my response, especially if it's, say, an autoimmune disease, will be, thank you, Captain Obvious. Because MHC should be related to any immunologic disease. If it's a disease that we never knew was related to the immune system, it's kind of interesting if MHC comes out as the relevant gene. But as you can see, we have infectious diseases, we have inflammatory diseases, we have autoimmune diseases. Basically, all of them are related to your specific MHC type. So the final thing that I want to tell you about today is a little bit of sort of, in some ways it's hypothesizing, but I'm actually going to give you some examples of what would happen to the human population if our MHC types were not diverse. So let's imagine for a second that we didn't have this awesome MHC diversity. What outcome, what bad things might happen to us as a human population? Yeah, Akuya. There might be some potential pathogen that could hit us all. It turns out that there are examples of this. So this is from when I went to South Africa the first year and when we went to cheetah rescue. Cheetahs have actually gone through a massive genetic bottleneck, and there is almost no MHC diversity in cheetahs. As a result, when you go pet cheetahs, you have to work on cleaning your shoes and cleaning your hands and doing a lot of things so you don't give them pathogens before you pet them. Because there is this big worry that cheetahs could have some pathogen that could wipe all of them out because they are so incredibly undiverse in their MHCs. So one reason why we're happy that our MHCs are diverse is because we cannot get this sickness like cheetahs. But the other reason why we are happy that our MHCs are diverse has to do with this animal. This animal is a Tasmanian devil. So before I tell you about the Tasmanian devil, I'm going to tell you something else. It's going to seem unrelated, but I promise you it's not. So let's imagine that Emilio has skin cancer. I'm sorry. And it's on his hand, okay? Now let's imagine he rubs his hand on Alina. Is Alina going to get skin cancer? No. Why you say no? Why no? Okay. So Molly says that's not how that works. Emilio says that's not how, Emilio said that the mutation hasn't happened to her cells. But why can't your cells grow immortally, indefinitely in her?
It's not the right environment.
The reason why they won't is because you have different MHC types. Her cells will actually reject yours as if it were a transplant. Because you have diverse MHCs, that cancer is not transmissible. And I picked a male and a female to make it super different with terms of antigens. And so in fact, if you took the immortalized cells that we work with in the lab, they are human immortalized cells, and you injected yourself with them, you would not get that cancer because you would probably have a different HLA type and you would probably reject them. If you happen to be super unlucky, that would be bad. But my other question is, why did you do that? So cancer is generally not transmissible in that way. Some cancers are caused by viruses, and the virus themselves could be transmissible, but cancer itself is not transmissible. So now back to the Tasmanian devil. Tasmanian devils, if you read their Wikipedia page, it tells you a lot about their life, and you will see the word biting frequently. It's like they just hang out biting each other in the face. I'm not kidding. The whole Wikipedia page is like biting, 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 biting. So they bite each other in the face. They also have gone through a genetic bottleneck and have very little MHC diversity. As a result, Tasmanian devils are quite likely to go extinct because there is a transmissible facial tumor that is being spread from Tasmanian devil to Tasmanian devil. Because their MHCs are alike, they do not reject this transmissible tumor from one to the other. And this may be driving these animals extinct because they like to bite each other in the face, and so they're rubbing their faces on each other, and that is actually transmitting the tumors one to the other. There are very few known transmissible tumors like this. It turns out that certain breeds of dogs are really inbred, And so there is starting to become a similar type of dog cancer in certain types of inbred dogs, it's causing the same thing. And then recently somebody found a second Tasmanian devil tumor because they are just that inbred and have that little MHC diversity. So if nothing else, you should be very excited that we have diverse MHC, because you are not going to all have to worry about the same pathogen like cheetahs, and you are not going to have transmissible tumors like the Tasmanian devils.